Okay, our next topic is restriction fragment length polymorphism. So far we have discussed about restriction enzymes, and restriction enzymes, as you all know, they are enzymes which uh, digest the foreign DNA that is coming inside a bacteria. We had a discussion over that. This is bacteria, they have their own genome, as well as when a virus infects bacteria, that virus sends its genetic material. So this restriction enzyme actually cut or digest the foreign DNA. This was the discussion we have done in the last class, and we also studied different types of restriction enzymes among the type 2, and we also had a discussion on restriction site. I hope you all are remembering this. Based on restriction enzymes, there is a technique which is known as restriction fragment length polymorphism. So today we are going to deal with restriction enzyme fragment polymorphism. Okay. So before starting that, you should know restriction enzymes, they cut at restriction sites and forms two types of ends. Let us take a DNA, and this is the restriction site of that particular restriction enzyme. Sometimes restriction enzyme makes a straight cut. That means this restriction enzyme makes a straight cut. When they cut in a straight cut, it forms two ends, and these two ends are known as blunt ends. Blunt. And this cut is known as straight cut. Straight cut. Straight cut forms always blunt end. The other way, restriction enzyme sometimes makes a staggered cut. For example, a restriction enzyme makes a cut like this. Okay, this is a staggered cut, or we can also show it as like this. And then it forms two ends, the ends will be like this. And such ends are known as cohesive ends. Cohesive. As well as the cut is known as staggered cut. Staggered cut forms cohesive ends. So please be careful while learning restriction enzymes. They make two cuts. One is straight cut and the other one is staggered cut. Okay. Okay, restriction fragment length polymorphism. This is a technique, and this technique used to compare two DNA fragments. And these restriction fragment length polymorphism is also one of the bases of DNA fingerprinting. Hope you all know about this DNA fingerprinting. DNA fingerprinting. This DNA fingerprinting is one of the very promising technique used in a variety of applications like forensic science, parental diagnosis, etc. Okay, let us first discuss Rflp, then move on to DNA fingerprinting. Rflp means if you take DNA and cut with restriction enzyme, they will form certain fragments. These fragments are formed which are unique to this DNA. That means हम एक DNA को अगर रेस्ट्रिक्शन एंजाइम से काटते हैं तो उससे जो फ्रैगमेंट बनता है वो फ्रैगमेंट का साइज, नंबर एंड ऑल इस DNA का यूनिकनेस है. मतलब ऐसा फ्रैगमेंट दूसरे DNA से नहीं बनेगा. Okay. The restriction sites would also vary, and the number of restriction sites also may vary between individuals. So if I take DNA from two individuals and cut with the same restriction enzyme, the fragments formed will be of different size and different number. और हम एक बार देखते हैं. That means कहना क्या है, एक इंडिविजुअल के डीएनए उसको आप एक रेस्ट्रिक्शन एंजाइम से यदि काटते हैं तो उसके जो फ्रैगमेंट्स जो बनता है वो फ्रैगमेंट उस इंडिविजुअल के लिए यूनिक है, बाकी किसी को भी वो फ्रैगमेंट वो पैटर्न नहीं मिलेगा. इसमें एक एग्जम्पशन है, लाइक ट्विंस. एज यू ऑल नो ट्विंस टू टाइप्स की ट्विंस है. एक ट्विन है एक ही जाइगोट से बने हुए टू इंडिविजुअल. दिस इज मोनोजाइगोटिक. तो मोनोजाइगोटिक ट्विंस को छोड़ के बाकी सबके जो डीएनए और उसके आरएफएलपी पैटर्न एक एक इंडिविजुअल के लिए यूनिक है. whereas मोनोजाइगोटिक ट्विन के दोनों का डीएनए का पैटर्न सेम रहेगा. Clear? Okay, we'll move on to the technique now. This technique says that if you cut individual DNA with a restriction enzyme and then you separate them based on their size, the size and number formed will be unique to an individual. Let us see, for example, I take DNA from two individuals, mine and my brother. Keep in your mind that we both are not identical twins. Okay, we are siblings. And if I cut this with same restriction enzyme are used for cutting this DNA as well as this DNA, and we get some fragments. Okay. Now these fragments need to be analyzed. So to analyze these fragments, we have a technique which is known as electrophoresis. It is also known as gel electrophoresis. That means DNA are allowed to move through a gel, semi-solid substance, and that DNA movement is based on two things. Suppose हम DNA को इधर डाल दिए, इस जगह को हम बोलते हैं well, and all the fragments are kept in this well. And as you all know, DNA is having some negative charge because they have phosphate. And if I connect this end to positive terminal of a battery and this to negative terminal, these all fragments will move through the gel. Now the speed of the DNA which is coming down depend upon its size and charge. All the DNA are negatively charged, then the speed of DNA coming downwards depend upon its size. So after some time we can see the DNA gets accumulated at different places, and these are known as bands. Now the one band contains multiple copies of same size of DNA. इसका मतलब क्या है, जो ऊपर के बैंड है, those DNA are heavier in size, and नीचे आते आते उसके DNA का साइज कम होते जा रहा है. ओके, आगे बढ़ते हैं हम. So what we have done so far, we have taken two individuals DNA and they are allowed to digest with the सेम रेस्ट्रिक्शन एंजाइम. फिर हमने उसके फ्रैगमेंट्स को एक जेल इलेक्ट्रोफोरेसिस के थ्रू सेपरेट कर रहे हैं. आई हैव लोडेड इंडिविजुअल ए, इंडिविजुअल बी, एंड देयर डीएनए आर लोडेड इन दिस well. Now we perform gel electrophoresis and we'll see what happens after some time. After some time we can see that certain bands are formed here, and these bands will have different pattern since the DNA have some variations. They may also have similar kind of bands, and they also have different dissimilar kind of bands. But altogether, if you take, this pattern is unique to individual B, and this pattern is unique to individual A. So Rflp help us to differentiate two individuals. As well as Rflp is also known as DNA fingerprint. Why it is known as fingerprint? Because this pattern is unique to one of the individuals. इसका बहुत सारे applications है, like parental diagnosis. Okay, another application है forensic science, and differentiating two species of two strains of bacteria. And all these things we can do Rflp. So Rflp, कैसे parental diagnosis, कैसे forensic, and how to differentiate two individual strains, let us see in detail. For example, हमारे पास एक केस आया जहाँ पे two individual claim कर रहे हैं on one child. We can take the DNA of child, mother, both the parents, or both the father, those who are claiming, P1 and P2. And we can perform Rflp, and their Rflp diagram will be like this. Mother and child will have some common DNA fragments. Parent 1 and parent 2. When we look through these DNA fragments, we can see that child's DNA or its fragments or its bands कितने मैच हो रहे हैं P1 से and P2 से. P1 से मैच हो रहे हैं 1, 2. P2 से मैच हो रहे हैं 1, 2, 3. So most likely P2 may be the parent of child. The same case or same way we can also or the same technique can also be used for forensic science. हमें क्राइम सीन से एक DNA मिला, उस DNA को हम Rflp किया. अब हमारे पास 10 सस्पेक्ट्स हैं, उन 10 के DNA फ्रैगमेंट्स को DNA आइसोलेट करके हम Rflp करेंगे और उस बाइंडिंग पैटर्न से चेक करेंगे किसका सबसे ज़्यादा क्राइम सीन से मैच हो रहा है. इफ़ इंडिविजुअल 1 इज गेटिंग मैच, तो उसका प्रेजेंस उस क्राइम सीन में था. दिस इज हाउ द DNA इज मैच, एंड DNA fingerprinting helps in criminology, in many other fields of science. So Rflp is one of the most important techniques, and the basis of Rflp is restriction digestion, and the different fragments formed from the Rflp is due to vary in number and position of restriction sites between two individuals' DNA. Okay, thank you.